Hi there, this is our another lecture about the fibrous protein. So in this lecture we are going to discuss about elastin protein. So elastin is also a fibrous protein, and this elastin protein forms the elastic fibers along with another protein which is fibrillin protein. Elastin protein forms elastic fibers along with a fibrillin protein which forms microfibrils, and these microfibrils act as a scaffold on which the elastin protein is deposited. How it occurs? Let us discuss it. Before going to the formation of these elastic fibers, we should be knowing the basic structure of elastin protein. Elastin protein is basically released in the form of tropoelastin molecules, which are the precursor of mature elastin protein. And this tropoelastin molecule has a polypeptide chain which have about 700 amino acid in its structure. And the principal amino acid found in this tropoelastin molecule include glycine, alanine, valine, proline, and lysine. Some amount of hydroxylysine and hydroxyproline are also found in it. However, the major amino acids are glycine, alanine, valine, proline, and lysine. So, how these elastic fibers are formed? These elastic fibers are formed in such a way that the cells, this is positively a cell here, it will release the elastin protein in the extracellular space. This is the elastin protein, and in extracellular space there is another protein which is fibrillin 1, which forms the microfibrils here. Supposedly this is the fibrillin 1 protein. It forms the scaffold on which this elastin protein is deposited. This elastin protein is deposited here, and it is surrounded by these microfibrils of fibrillin 1 protein. So, in the center there is elastin protein, and on the outside there is fibrillin 1 protein. So, there is also fibrillin 2 protein, but fibrillin 1 protein is in a major amount. So, in this way the structure of elastic fiber is formed by fibrillin protein and elastin protein. Then when they are deposited here, then the tropoelastin molecules undergo cross linking. They undergo cross linking which imparts them the greater strength and imparts them the rubbery nature. So these elastic fibers, due to presence of this elastin protein, they can be stretched to the longer lengths, and then they shrink back to their original length. So they impart elasticity to the tissues. So they are found in the blood vessel, they are found in the elastic ligaments, and so on. Another thing we should be knowing here, that the major linkage here which occurs in the elastic fibers is desmosine linkage. Desmosine linkage. What happens? The lysine residues present in the elastin fibers are oxidized. Lysine are oxidized to L-lysine. Previously discussed in the structure of collagen as well. They are oxidized to L-lysine molecule, and three L-lysine molecule join with one lysine residue. They join with one lysine residue. Three L-lysine molecule join with one lysine residue and form a desmosine linkage, which is a complex bonding which imparts the high strength and elasticity to these elastic fibers. So, another disease we should be knowing here in context of these elastic fibers is the Marfan syndrome. Marfan syndrome. So, what happens in Marfan syndrome? In Marfan syndrome, the fibrillin 1 protein is defective. Fibrillin 1 protein is defective due to genetic mutations, and when this protein is defective, then the elastic fibers are not synthesized normally. So, what happens? It leads to the cardiovascular symptoms. For example, there is aortic aneurysm, there is aortic aneurysm, there is aortic dissection. It leads to the ocular symptom. For example, there is myopia due to lengthening of the eyeball. There is ectopia lentis due to dislocation of the eye lens, and so on. And there is also skeletal changes. The patient is tall and also have tall thin limbs, large or thin limbs, and the patient has blue sclera due to thinning of the connective tissue of the eyes. So, the elastin fibers, which are synthesized by a combination of fibrillin protein, this is fibrillin protein, and in the center there is tropoelastin, which is highly cross linked, and this is the elastic fiber. When this fibrillin protein becomes defective, Then the Marfan syndrome occurs, due to which changes occur in the cardiovascular system in the form of aortic aneurysm, aortic dissection. Then there are symptoms in the eyes in the form of myopia due to lengthening of eyeball. There is the ectopia lentis, or dislocation of eye lens, and there is blue sclera due to thinning of the eye tissues. So it is about the structure of elastin and how the mutation of topoelastin affect the elastic fibers and tissues of the body. Next, we should be knowing that the elastic fibers are also present in the elastic tissue is present in the lungs. Elastic tissue or elastic fibers are present in chief amount in the lungs, because lungs is an expandable tissue which expands on inspiration and contracts on the expiration. So it is rich in the elastic fibers. So what happens? There is enzyme which is called neutrophil elastase. It is present in the lungs and released by WBCs there. So this neutrophil elastase has the tendency to digest the elastic fibers of the lungs and can damage the lungs. So this neutrophil elastase is inhibited by enzyme called alpha 1 antitrypsin. This alpha 1 antitrypsin is basically anti-protease or protease inhibitor, which inhibits this neutrophil elastase and protects the lung tissue from the damaging effect of the neutrophil elastase. However, what happens in smokers? In smokers, there is the deficiency of alpha 1 antitrypsin. How? Because the smoking oxidizes the methionine. This alpha 1 antitrypsin has the methionine at the 358th position. At 358th position of its polypeptide chain, there is a methionine which helps the binding of antitrypsin to this elastase. However, the smoking oxidizes this methionine and forms methionine sulfoxide. Smoking forms methionine sulfoxide, which cannot bind the neutrophil elastase. So as a result of this oxidation, alpha 1 antitrypsin does not bind to neutrophil elastase, and when it does not bind, it cannot inhibit it. So smoking leads to the destruction of alveoli due to protease action of this enzyme. As a result, there is destruction of alveoli and there is the disease which called emphysema. So in this way, smoking can lead to the emphysema. Another thing we should be knowing that there can also be the genetic deficiency of alpha 1 antitrypsin due to genetic mutations, and if it arises due to genetic deficiency, then what happens, the mutated alpha 1 antitrypsin can accumulate in the hepatocytes, because principally alpha 1 antitrypsin is synthesized by hepatocytes and released into the blood. So what happens, mutated alpha 1 antitrypsin accumulates in the hepatocyte and damages the hepatocyte, which can lead to the liver failure or liver cirrhosis. And this condition arises when the patient is homozygous for the defective enzyme, or patient have both the alleles defective, or both the genes for alpha 1 antitrypsin are defective. However, if the person is heterozygous or have one defective gene, then the one normal gene synthesizes enough alpha 1 antitrypsin that he does not develop symptoms. However, if this heterozygous patient smokes, then the alpha 1 antitrypsin further becomes deficient due to oxidation of methionine, and then this patient has the very increased risk of the developing the emphysema due to alpha 1 antitrypsin deficiency. So the principal mutation which occurs in the defects of alpha 1 antitrypsin is the replacement of the GAG code with AAG code, and when this code occurs, there is change of lysine in place of glutamate. So glutamate at the position of 342 is replaced by lysine. So the mutation occurring in the genetic defect of the alpha 1 antitrypsin is GAG code is replaced by AAG. So at 342 position, glutamate is replaced by lysine. As a result, the mutated protein is produced, which accumulates in the hepatocyte, can lead to the scarring of the liver. However, the individual must have two abnormal alleles for the development of emphysema. If one allele is defective and one allele is normal, then the patient have the enough level of alpha 1 antitrypsin that the clinical sign and symptom does not develop. However, if the person smokes, then the typical symptom of alpha 1 antitrypsin arise and patient develops emphysema. So it is how the elastin molecules are arranged into the elastic fibers, and how fibrillin 1 deficiency leads to the Marfan syndrome, and then how the elastic protein or elastic fibers are degraded in the lungs by neutrophil elastase enzyme, and how it is prevented by alpha 1 antitrypsin, and how alpha 1 antitrypsin can lead to the development of emphysema. So it was all about today's lecture. Thank you.